So welcome everybody to the disappointment of our webinar Talking Trial Science. And my name is Sorja Bolocka from CAT Europe, and I will chat today with Silke Keller from the Trial Center Tübingen. And we are really happy to have as our guest Fenna Cilliers from Johns Hopkins University, CAT. Then I'm really happy to introduce you, Fenna. And it was very nice in the bio, starting immunologist with a passion for environmental health. So I think it's a very nice subtitle. So Fenna, before joining Johns Hopkins, she received a PhD in immunology at the Utrecht University, and she did her work at the Brigham Women's Hospital in Boston. And then she has done a postdoctoral fellowship in functional genomics and environmental health at the University of California in Berkeley. And now she's an assistant professorship at Johns Hopkins, and she's in the environmental health and engineering department. And she's researching on early life exposure change of the immune system and increase the risk of respiratory infections and lung cancer later in life. She's not only assistant professor, she's also deputy director of CAT since 2021. And today she's here in the role of director of the Johns Hopkins Exposome Collaborative Program since 2019, where she oversees several projects, including the Baltimore Asthma Exposome that I'm sure we will hear about today. And so I'm very happy, Fenna, they accepted the invitation, and please, the floor is yours.
Yeah, well, thank you so much for inviting me and to speak at this webinar. It's really an honor, and I will start my presentation. Does everything look good? Okay, cool. So yes, thank you for the introduction. That means that I don't have to introduce myself anymore. But I am Fenna Cilliers, and I'll be talking to you today about the exposome, a lifetime of exposures complementing the genome. And I will do this at a broader, higher bird's eye view level so that you would understand what the concept means, what the tools are that we use to assess the exposome, and the type of applications that we can apply the exposome paradigm to. And then for details on the nitty-gritty, I would advise you to contact me or anybody in the field for more follow-up. But this is a bird's eye view to introduce the concept. So first, a quick poll, because I would like to know who of you knows what the exposome is. So it's a good mix. That's good. So 36% of you know what the exposome is. I hope you still get away with some new knowledge or insights on the exposome after this presentation. 38% of you did not know, and I'm excited about that because I hope that after this presentation, you do have a grasp of the general concepts about exposome and what we can use it for. And if you're not sure now, hopefully after this, you will be a bit more sure about what it means and how you can implement it or use it in your own research, maybe. So I'm going to close this poll and introduce the concept, the paradigm. So the exposome was coined as a term in 2005 by Christopher Wilde, who is the former director of the International Agency for Research on Cancer, IARC. And in this article, he basically indicated that we needed to, as a field, complement the genome with an exposome. And that is an outstanding challenge because there is a plethora of environmental exposures that need to be measured and can be measured in molecular epidemiology, which is the study of health and disease. And he wanted it really to be at the level of how the human genome was assessed and queried. We really need to complement that with an environmental component. And the reason for that is what I'll go into a bit more detail, is because it turns out that not all diseases are genetically linked. So the concept of the exposome paradigm encompasses the totality of human environmental exposures. And with the totality, we mean both internal and external. So what do we mean with internal and external? With external, it's very obvious. It's the air you breathe, the water you drink, everything outside of your body that can be measured and has an impact on you. But it also means what is internally. Because internally, when you ingest something, the easiest example is, for example, food. If you ingest food, You metabolize it, you break it down, your cells eventually take up some components that were broken down by your gastric system and use those components for their own cellular metabolism, for their own energy, and change that again. And your cells themselves then excrete other factors as well, signaling molecules, metabolites from components that they have been using in their pathways. And that then gets released to the microenvironment around at the cell level, basically. And that is also something that other cells, neighboring cells, are exposed to. So really, in a sense, that exposome does not just capture everything that's outside, but also all the processes that happen inside. And a lot of these processes inside are a reaction to what you are exposed to from the outside. So there is that interplay. It means even, for example, the microbiome, which is part of our host system, they're cranking out a lot of metabolites, a lot of factors as well that we are exposed to internally. And that can differ at either the population level, because air pollution in a city, the whole population is exposed to that. But at the individual level, you have different levels of exposures as well. For example, everybody's microbiome is slightly different. So you have slightly different exposure internally. Another critical element of the concept is that it was initially coined as from conception through death, basically a whole lifetime. We're now actually refining, and I'll show you a few waves of refinement of this concept, or the original concept, how Christopher Wilde put it out. But it's not just from conception anymore, is what we're realizing. It's even before that, because gametes and oocytes all are already existing prior to that. They come together and make an embryo. So even your maternal exposures prior to conception are important, and same goes paternal exposures, because as you'll see later on in the talk, an external exposure, for example, can leave an imprint on the genetic blueprint of different cell types, and then when they go together during conception, that imprint is taken along and is carried forward in the organism. It's really a lifetime, and you could even extrapolate to a transgenerational lifetime of exposures, both external as internal, that interact with the genome. And it's really complementing the genome, because you have your genetic blueprint, that is who you are by nature, and that blueprint, that genomic blueprint, interacts with anything else. You as a person interacts with the environment, you as a person have internal processes that you're exposed to. And it's not just the external, it's not just the genomics, it's that interaction. So a lot of little pieces of how you should tease apart this one-liner concept, but it's critical to realize that interaction. So as I mentioned, over time, so Christopher Wilde coined the first time exposome in 2005. And then there was not too much follow-up, but in 2010, Steve Rappaport and Martin Smith picked up on the idea and basically evolved it more, made it more practical, redefined a little bit that the exposome is all exposures from conception onward, including those from lifestyle, diet, and the environment. So not just environmental components, because the environment is not just the external environment as you think of the milieu, but it's also the choices you make in that space. So your lifestyle, what you eat, what you do you exercise, or do you only drive your car and not walk, things like that. So all those factors also have an effect on your biological processes. So what they also did, which was very important, is they made it very practical, because how do you measure the exposome? You can't go out and measure everything external and everything internal at this moment yet. But what you could do is look at internal chemistry, internal processes. And because there is an interaction of the external environment on the internal environment, you can actually assess the exposome in blood samples from somebody. So basically, it considers that the external or any type of exposure, even internal exposures, leave an imprint on either cell types or DNA, certain proteins, like as adducts, or the function of cells can change, and therefore the analytes that they crank out during metabolism or something like that will change. So that is a profile that you can measure, it's an imprint that you can measure as the result of your exposures. So even though you can't measure every chemical, every event in your lifetime and prior lifetime, so even though you may have a history of unknowns, you can still measure the exposome by the results that it is at the time of assessment. So basically, your health status is a reflection of everything that happened in your lifetime. And you can assess that health status, that profile, that imprint, without knowing all the details from your history, is basically. So they made it very much more practical to measure and go about assessing the exposure. Another wave of refinement came from Gary Miller and Dean Jones, where they indicated that it's an cumulative measure of both environmental influences and associated biological processes. So it's not just the imprint, but it's also the response to. And that comes more to the, for example, the metabolites, the function of the cell that changes if you have a history of X exposures. Your cells even at this stage in the future will behave according to the experience that they have seen in the past. So, and again, they acknowledge, they take into consideration that broader concept of it's the environment, including diet, behavior, and endogenous processes. So the concept of exposome has been redefined a little bit over time, but at the moment, it's really a lifetime of exposures, which happens already pre-conception, so at the maternal paternal level already, of any environmental influences that includes biological responses. It also includes the environment, diet, lifestyle, social economic status, psychosocial stress. All these factors and all these events in your past contribute to the exposome. So why do we care about the exposome? Why was it even necessary to think beyond the genome? Well, there's always a Lancet report where every 5 years or so, where the global burden of disease is assessed. And that's a collaboration of multiple countries where they look at the mortality rates by disease. And then you can see that shifts over time. So in the 1990s, ischemic heart disease, stroke, COPD, and infectious diseases such as tuberculosis were in the top 5 causes of death for, in this case, I highlighted the later adults, so 50 to 74 years old. And this has changed since then. And now in 2019, which is the most recent report, ischemic heart disease is still, and stroke is still in the top. COPD lowered a little bit, but diabetes and lung cancer are also still there. So tuberculosis and diarrheal diseases, you can see, really drop down in the list. So we're better at protecting our populations against infectious diseases, pathogens, but these chronic diseases are in the top. And the whole world is catching up on that. So it's very Western, is what they sometimes call them, diseases. But these are diseases that are not due to a pathogen. It's really chronic disease. And what is notable is that these are not only caused by genetic factors. It's definitely, these have all been shown to also be impacted by environmental exposures, by external exposures. That leads me to a poll. What percent do you think the genome accounts for in the chronic disease mortality? So you voted all over the board, which is good. I like that the responses are different because some know per disease, some may know from the general reports. So I'll show you. It is different per disease type, but overall, there are some numbers that really indicate that we should be looking at the exposome. And I'll show you that right now. In order to study this, like how much comes from the environment and how much comes from genetics, most of this data comes from monozygous twin studies or adoption studies, but the twin studies are the most notable. And in 2015, there was a meta-analysis done of the heritability of human traits based on 50 years of twin studies. And it was a massive meta-analysis because they looked at more than 17,000 traits from more than 2,748 publications. And within those publications, within those studies, a total of 14.5 million twin pairs were included. And what they found was that across all traits, the reported heritability is 50%. That's any trait, like hair color, eye color, things like that. 50% is heritable. But that doesn't obviously tell us about disease status. So there are other studies that have teased out a bit more by disease type what the contribution of the environment is. So for traits, 50%, more or less 49 versus 51 is very, very 50-50. So an important part of our traits are determined by the environment. But for diseases, Dr. Rappaport had done an analysis where he looked at Lancet data of those global burden of disease. And he basically used the concept of population attributable fractions. So that's the path, the percentage there. And the population attributable fraction is how much of a disease is attributable to genetics. If you could solve the genetics, how much percent of the disease mortality would you reduce in that population? And as you can see, for ischemic heart disease, this translates about to 21%, and then lung cancer 9%, and for pancreatic cancer, for example, only 4%. So the genetic contribution to most chronic diseases that are in the top 10 for global mortality rates are not fully dictated by the genome. So between 70 and 97% of it is due to the environmental factors. So this is what drives home the need to look at environmental exposures. And because we don't live in a vacuum, we don't only expose ourselves to one metal or to one air component or just water as it is. No, there's also salts in the water. There's also metals, other things in the water. Everything around us is a mixture, and therefore we should consider that as the exposome. It's not a single exposure that we need to, in the field, go after to study. So he went on more to look also at the Lancet report to tease out more, because what they do also have a bit of a disease burden and a disease mortality. So this is why it's very important to study the environment and the components that are contributing to disease, and to study that not in a single silo, but to study that in a holistic manner through the exposome. So another idea here is, and this slide is adapted from Thomas Hartung, who is the director at the Center of Alternatives to Animal Testing, is that the disease is driven by 3 main components, and that's represented in this triangle. You have your host component, which is obviously the genetics, your genome. You have a pathogenic component, so pathogens contributing to disease, and you have your environment. And more than 40 years of the human genome, since the human genome has been sequenced, we found in the field that about 40% of all the 560 diseases that they looked at in this paper had a genetic component. So very similar to that traits, the twin study traits, like 40 to 50% is genetic, but that means that there's 60% of diseases that have no genetic component whatsoever, and thus either are driven by the pathogen or by the environment. And the Rappaport and Smith have indicated that this is about 70 to 90% is what they calculated. And we already showed that in the previous slide. So over the years, over those 40 years, the world spent more than 3 billion on the Human Genome Project. And that's not counting all the sub efforts. And the Patel report has indicated that between 1988 and 2010, human genome sequencing projects, associated research and industry activity, and directly and indirectly have generated an economic output of 965 billion, with a personal income exceeding 293 billion and 4.3 billion in job years of employment. So a lot of time, a lot of money has been spent to the genome. However, we already showed that a lot of these diseases are not fully genetically driven. So there's a very discrepancy between how much the contribution is of the environment and how much money is spent on the genetics versus the environment. Another person who is very active in the field of exposome is Gary Miller, and he has 2 books out, The Exposome and The Expose a Primer, and This Exposome, A New Paradigm for Environmental Health. And in those, he has nicely described the exposome concept, how it developed, but also framed it into the broader context. And a quote is that he mentioned that it's odd that the majority of the discussion of Darwin, which reflects the money that we spend on the human genome, occurs amongst geneticists when natural selection is primarily driven by the environment. The environment is the exposome is what is driving those natural selection. If you remove it, if you remove any of these external forces acting upon our genome, evolution would grind to a halt. So again, another logic of that, the exposome and looking at environmental components in interaction with your genome. So we don't want to ignore the genome, but in interacting with the genome is quite important. Again, there are obviously some diseases that are fully genetically underpinned and or have a big component, genetic component, but the majority of the diseases, and at least 50% of the diseases, has no genetic component, is environmentally driven. So we had a Johns Hopkins Exposome Collaborative here at Johns Hopkins. We put out a food for thought article. It's called The Exposome, A New Approach for Risk Assessment. I encourage you to look that up. But we have put some frameworks and some challenges in the field forward that need to be tackled. Because even though the paradigm was conceived in 2005, there's a lot of discrepancies, sort of the refinements of the paradigm and the concept itself, but also the challenges of how do you implement and how you actually assess the exposome are being discussed here. So overall, if you want to study the etiology of diseases, you look for the genetic component, which obviously drives the susceptibility. And then you look at the interaction with either the infection, including the microbiome, and that also dictates your susceptibility. And then the exposure, including physical and social, that interaction with your genetics also drives disease. So there's a genomic component and the exposome that there's a balance and an interaction that happens, we would need to study. And if you look at it from trickle-down processes, it's the internal environment that in the individual level, you can measure at the body fluids, and that you can call those imprints, you can measure the exposome in that way. And that internal environment in a person is affected by your lifestyle, specific external environment. It's also affected by your general external environment, and that's unavoidable exposure sometimes. I mean, these are choices. This sometimes is not a choice. Where you live or where you grow up is sometimes not a choice. And then these factors in the internal environment can have an influence on the epigenetics, as I said. They can leave imprints at the genome level, which then dictate whether certain genes are transcribed or not. That has influence down the road in that sense. Genetics plays a role, because if you have certain SNPs or gene variations, that definitely also plays a role in how you, for example, metabolize things. So that at the individual level also plays a role and interacts. Your disease history, especially if it's pathogenic diseases, but even if you had diabetes, you cured yourself, that leaves an imprint as well. And then at the individual status, the outcome is your health. And your health could be monitored by biomarkers and personalized medicine, and external exposures and effect of relationships and regulations feedback into how you behave as an individual, etc. And as an individual, you're part of the population. So Exposome even touches on public health. So what are the tools to assess the exposome? As I mentioned, the holistic concept of the exposome is both external as well as internal. So there's tools that you can use to measure the external exposures. For example, wearable devices, smartphone or accelerometer, environmental sensors for air pollution or anything else, water quality testing. A lot is done also by questionnaires. So do you live in a green area? How many meters do you live away from a major highway? All these things at the regular census data to very detailed questionnaires can also capture what has been going on externally. A lot of ways to measure external exposures is also by geospatial environmental modeling. For example, air pollution above certain regions, here in this case in France, or green space, noise, things like that can be modeled either by satellite data or by measurements from governmental trackers that are placed at strategic locations throughout the country, for example, to monitor air pollution, things like that. There's a lot of environmental modeling that you can implement also to assess the external exposome. From dietary and what you are exposed to during your lifestyle, you can learn a lot from pictures that are made from the bottles and the ingredients that you are exposing your shampoo or your drinks and things like that, cleaning products, your food. You can derive a lot of exposure from those. And then internally, you can look at biospecimens. Urine is a very easy biospecimen to give at the individual level. Blood is relatively easy. I mean, it's invasive. It's doable in that sense. Exhaled breath, condensate. Sometimes people do exercises on a treadmill or something like that to measure and then measure cardiac rhythms, things like that. Those are all things you can measure. Some of these can result into biomarkers, and that could then predict a disease, or it could predict or indicate what type of exposure you've been exposed to. So it depends on what type of biomarker you're looking for, a biomarker of exposure or a biomarker of the disease that is going on. And then with omics being high-level technologies that look across and very untargetedly at, for example, the proteome, looks at all the proteins in your specimen, in your biospecimen. The epigenomics looks at DNA methylation, histomodifications, etc., all at the whole genome wide. Transcriptomics is the RNA expression. Proteomics, metabolomics is, for example, what cells secrete after they have metabolized and processed, for example, components that they have been either exposed to or normal processes in the body that release analytes and release metabolites that at the internal level you could be exposed to. So those can all be measured either, for example, by sequencing, mass spectrometry. And another interesting one also is that reactive molecules can react with big proteins or with DNA in the body and basically get stuck and form adducts is what it's called, and leave an imprint that way, which you can measure. And then that imprint becomes a measure of what you have been exposed to. And especially at the level of DNA and protein, certain proteins are stable for 30 days. So if you have repeated measures, you can infer over time how much you've been exposed to at certain time points in your life, and that you can then make an inference of what your exposure has been over your lifetime. So different ways of measuring the exposome. So how many of you already use multi-omics or multi-assessment approaches in your research? Because traditionally research is like one chemical, one outcome, or one chemical, a few outcomes, but it's not that mixture. It's not that looking at a holistic, broad level of what your exposome would be like. So how many of you have done that already? And it doesn't have to be everything. I'm just curious if you, for example, have combined transcriptomics with proteomics, because the nice thing there is that you have at the RNA level what changes. But does that really mean that there are changes down the line in your protein concentrations or in the expression of certain receptors? So it's very nice to pair multi-omics because it gives you also a sequence of events that you can then determine. Another way to approach multi-omics is that multiple ways of exposure, you capture different types of exposures or different imprints with different methods. So, wow, it's really a good mix, 50-50, whether people are using a multi-omics or not. Now, I'm not saying that you have to switch to multi-omics approaches, but now you can consider that if you ever want to explore further in your research, there are ways to also integrate and look at a more broader level of how your individual exposure or toxicant that you are interested in is playing a role in exposome picture in that sense. Thank you for filling out all these polls, by the way, and being so interactive with me. So in those papers, we have put forward a way of approaching the exposome, and you could do that with the human whole genome expression. So transcriptomics, pair that, for example, with untargeted metabolomics at whatever level, and then you get to a pathway of toxicity. And you can implement systems biology and bioinformatics to tease out what the molecular pathways are. And you can do the same for the metabolomics and then find what the metabolomic pathways are, because this will give you a little bit more depth. So not just what is the exposure, but also what are the pathways that are changing once you've been exposed to your exposome. And then you can do cross-omics analysis, for example, in vitro, where you use human samples, and then you measure the validation of any of these mRNA pathway targets that you discovered. You can do targeted metabolomics to follow up on any pathways that you identified with the untargeted methods here, targeted proteomics, etc. All these down the line steps to come to a sequence of events that lead to eventually an outcome, an adverse outcome, because often we are studying the exposome in the context of disease. So these methods will help you get anchoring your phenotype or find what the biomarker of effect is. So now switching gear to applications for exposome studies, it would be really ideal, and I'll touch base on that at the end of the talk, to have a lifetime of exposures, whether you're healthy or not, and know everything what you've been exposed to. The hard thing is that we don't know how to measure everything. We have imprints that we can measure, but it's also hard to tie that to an outcome if you don't really have anything changing. So the exposome of a healthy person is almost harder to detect than the exposome of a disease. So most exposome applications and studies out there are actually looking at the exposome in relation to a specific disease, even though in your lifetime you may be exposed or you may get multiple diseases. We still study that as a field in a silo per disease. So a lot of people, for example, look at the asthma exposome, and our Hopkins collaboration does that as well. So external, there are individual factors that you expose yourself to, and there are broader environmental factors that leave an imprint and interact with the internal environment and then lead to health risk. And that is something that you could study in an exposome study. So how does the exposome, and you can have that with or without interaction with the genome, drive health risk of, in this case, asthma. Another example is that people are looking at the exposome at a more environmental level. So, for example, they focus on if climate change happens, what type of exposome is associated with that? Because climate change also doesn't occur in a vacuum. It will change multiple processes and events throughout the world that are impacting our life, that are forming our exposure, our exposome. So that's another approach how you could tackle studying the exposome is by events in the environment. So climate change in this case could lead to increased use of pesticides, leakage of chemicals due to the deterioration of containment, temperature increases, certain vectors can be going to places where they normally don't exist. There's an increase of exposures through, for example, the pHs that are released due to energy use, etc. So these all have impact on either cancer, health, bacterial infections, etc. So this is another way how you could tackle the exposome and integrate the exposome into your research. Another very interesting thing is because a lot of these previous studies is more also at the population level. So climate change, how does that change people's exposome, but at the population level? Another interesting level is at the individual level. So you could also use the exposome for personal precision medicine. And one of the very early, although they didn't call it the exposome, studies was by Michael Snyder, who basically did any omics that and any measurement that he could think of on his cell for over a year with multiple time points. And he profiled all these omics, all these measurements, and found, for example, that he was pre-diabetic, did an intervention, and that was the pre-diabetic was picked up by, I think it was the metabolomics, did an intervention, then became into a health status that was not pre-diabetic anymore. So he cured himself from imminent diabetes, and that would have been happening if he hadn't monitored himself. And there were a few other things that they picked up on during an infection, things like that. But really, it was one of the first examples of how you could monitor at an individual level and then tailor interventions based on these omics, because they interact and then predicted much better what health status he was at the time of measurements. So precision medicine is very much looking at incorporating the exposome and having it interact with the genome. I just added here that he could have measured even more things, but aside from that, they did a very extensive profiling on in this case, just one person, but you could do this at the level of the population, but also at the level of an individual for precision medicine's sake. And precision medicine, as I mentioned, is not just measuring everything that every omic that you can think of that is external, but also how these genes interact with the exposome. So genetic variations from GWAS studies, how they interact with the exposome could lead to better risk prediction of autoimmune diseases, inflammatory disorders, and cancers, for example. So a lot of these non-pathogenic diseases can be predicted or have better resolution if you do a gene times exposome approach. So really quickly about our exposome collaborative at Johns Hopkins. It was founded upon funding from Ji Wu and Xiaomai Chen. And our goal is to characterize the cumulative imprint of environmental influences and associated biological responses throughout the life course, and do this in a holistic fashion, looking at the environment, diet, behavior, socioeconomic factors, and the endogenous processes. But we also focus on developing new tools, integrated analysis, best practices for statistical modeling, building a framework to address both ethical as well as legal and social structures. And we do that through a pilot on the Baltimore exposome, the asthma exposome in childhood asthma in this case, where we're leveraging an ongoing study which already is measuring a lot of environmental components, and we are adding all these additional omics, all the additional assessments on there to really get a holistic profiling of what the exposome could be. So basically a model pilot study. And as you can see here, we're really trying to tackle all different levels of the exposome. So far, most studies have only done one omic with one maybe exposure, or just an omic with a survey data, things like that. But in theory, you want to look at everything, every level, to make sure that you have the most complete exposome picture possible, which is a challenge and will cost a lot of money. So I think similar as to the level of that we spent billions and billions on the human genome, I think the research field in general should start thinking about how we can really thoroughly measure the exposome by dedicating funds to do this in a more holistic manner and not on a one-by-one omic case. So we put out a framework where we are looking at both at the public health as well as the individual level for external exposures that also interact with the genome. But we also definitely focus on psychosocial stressors. We define chemical stressors, and there are community level stressors that also influence the individual, and the community level exposome. We will measure biospecimens and all those omics to get an imprint. And with a lot of big data, we come challenges. So we have proposed also that we would need to employ AI techniques and modeling to understand and process this, integrate all this data. What we acknowledge is that when you go at it in an untargeted fashion and with so many different omics at the same time, you may generate hypotheses that you can then test in a targeted way. And that was the framework that I earlier explained from the paper that we put out. We also acknowledge that public health is driven by the individual as well as the community level exposures. And what we realized also is that there are bioethical concerns with the exposome, and I'll get to that a little bit in a second, but we want that community also to be part of how we do research on the exposome, because this is a new field, and similar as the genome, the exposome may very well serve as some sort of imprint or fingerprint of who you are. So we need to include the community and the bioethicists from the beginning before we do this at a massive scale. And as I mentioned, we're looking at AI, exposome intelligence also, to help us with analysis of big data. So there are challenges with the exposome, and I mentioned that we need new technologies because we are not measuring everything yet. What we don't know, we don't know. So there's a lot of unknowns. So, but as technologies improve, we may detect at lower sensitivity chemicals, or we may detect new compounds that we haven't been able to detect with the current methods and things like that, or new imprints that we haven't been as sensitively being measuring. So there are challenges there that will be addressed in time. Infrastructure is needed. So there's not a lot of standardization. Everybody in the field at this moment is doing the exposome analysis with whatever they can afford. A few omics, maybe an external exposure or a survey census or whatever. So there needs to be more harmonization and best practices. We need to come to using the same terms, like not everybody still defines the exposome the same way as everybody else. Databases, this big data being generated, we need to store that. We need to be able to integrate that. So a lot of challenges there also. And then, as I mentioned, there are ethics involved. So can we use existing biospecimens for exposome research? Do we need to reconsent? What do we do with data, exposome data, when we store that in the future, because it may be an imprint? So these things are things that we need to consider as a field. And eventually the exposome is about integrating all this, making connections. And as I indicated, there are many diseases that can be studied and have an external component. So studying the exposome in that context makes sense. And there are efforts around the world already where big funding has been spent, where at the Exposome level, research is possible. In Europe, in the US, and in Japan are examples of these ongoing processes. And these are just, it's not a complete overview, but these are the major funded projects that are out there that could do Exposome level research. And then for the future, maybe there is a possibility that we can have a human exposome project, similar as a human genome project, where we determine in a very holistic way what the exposome would look like from an individual at a population, but also at the individual level, and what the standards would look like there. So that's one of the efforts that have been highlighted by a workshop by the Secretary of Defense and Research from a workshop from last year. So this is definitely a big undertaking, and I am excited to follow up on this, to follow this field. So with that, there's this call for the Human Exposome Project, and for exposure-driven, technology-enabled, and evidence-integrated research about the exposome. So with that, I would like to conclude. If there are any questions, please put them in the chat or post them so that I can answer them for you, hopefully. And I hope you enjoyed this talk, that you learned about what the exposome is. And if you already knew, that you learned a few new things of how to implement it or tools that you could use. And if you have any questions that I'm not able to address here during this webinar, feel free to reach out, contact me. So thank you.
Thank you, Fenna. This was really, really comprehensive. It was a nice overview. And we have just a few minutes for the discussion. We went a bit long, but we would like to take a few questions if there is time. So you can write in the Q&A or in the chat. And of course, you can, I suppose, you can contact Fenna or the Exposome project. Yeah, there are all the general email. So let me see. Well, we're just congratulations on your talk in the chat. Maybe Fenna, I have a question.
Yeah.
I mean, of course, it's everything complex because we are a complex system in a complex world, and indeed, how to deal with all these variables. So it's really the AI, the solution, because at the end, it's really already in a small system like a cell-based, you still have a lot of things that can influence it. And when you see the reality, it's like, okay, oh my God, I will not.
Yeah. No, for sure that is one of the challenges. You can't measure everything. Also, there's a few things that you could measure in the past. For example, there may be records of air pollution, things like that. But that's where the power of the omics come in, where, as I mentioned in the beginning, your health status at this point is a reflection of what happened before. So even though we don't know exactly what you were exposed to, we don't know what the outcome is, what the imprint that left. And that is a sort of substitute, an imprint that you can use to assess whether you have been exposed or, so you could compare similar profiles from a known exposure and then infer whether that type of exposure has occurred, or even if it remains unknown, it is a reflection of what happened in the past. So that's why it's important to combine omics with external measures, because the omics comes close to touching on, well, even though we don't know that, we can still know a lot. But methods will improve. I mean, mass spectrometry over the past 10 years improved significantly. So we're now detecting levels of chemicals and compounds at much lower sensitivity. So we're now able to measure things that we never knew were there. So I think improvements in technology will happen in the future and will aid. And then, yes, with all this data, the big data, but also the knowledge of machine learning, you can also train models on those gaps, those information gaps, and have the machine learning do inferences on those gaps. So that will get better over time as well. I mean, it's very pioneering right now, but the more data we will generate as globally on these data sets, on bigger data sets, the more we can also have these models train to make inferences and fill in those gaps without actually measuring it, but by imputation or whatever. Yes. Yeah.
Okay, sounds exciting.
Yeah.
Okay. Then thank you again, Fenna. This was really, really interesting.
Yeah, thank you for inviting me, and I'm happy that you were willing to listen, all of you.
Yes. Then have a nice day, have a nice evening,
Thank
and
you.
see you next time. Bye-bye.
Yes. Bye-bye, everyone.